Good morning. Sorry I can't be with you this morning. I've got a medical appointment I can't get out of. I can't be there in person. I've got a medical appointment later in the morning. But let me kick us off now with a talk about adventures with metagenomics. Rather fanciful title, but you're going to learn how to analyse metagenomes this week, and I'm going to provide some conceptual background and look at a case study for my own research that you're going to be drawing on in terms of your data set. So when we're looking at clinical samples or environmental samples and we want to know what's in them in the form of bacteria, we've got a number of ways in which we can move forward. One approach that we mustn't forget is that you can actually grow bacteria in pure culture from such samples, and this has gained speed, adrenaline, force, if you like, in recent years with the advent of something that's called culturomics. It's a rather ugly neologism, but it captures the idea that with high throughput culture methods and high throughput sequencing, you can actually get an awful lot of information. However, when we're looking at complex communities, many of the bacteria and fungi and other organisms, and particularly the viruses, you can't actually grow them very easily in the lab, and that's why we extract DNA from these samples and analyse that as a way of trying to work out what's in there rather than growing bacteria. Traditionally, for decades now, there's been an approach where we amplify fragments of the 16S ribosomal RNA gene, do PCR on that. The reason this works is that there are conserved parts of that gene which flank variable regions, and so you can use this as a kind of molecular barcode. We're not going to say much about that this week because it's a rather old-fashioned technology, and the future belongs to metagenomics, what some people call shotgun metagenomics, to differentiate it from 16S. But it's this what we're going to focus on this week. And as with when you're doing genome sequencing of cultured isolates, here you make a sequencing library, you do high throughput sequencing and then assembly, and then you can do further analyses. So here, in a sense, instead of doing all the heavy lifting by trying to culture loads of different things, we do the heavy lifting at the stage of the bioinformatics analysis of the sequences that we've created. If you like, it's a massive, huge, complex jigsaw puzzle that we have to solve. Here's another slide, summary slide that summarizes the same thing. You can do culturomics, you can extract DNA from metagenome sequencing. Once you're doing metagenome sequencing, though, there are two different approaches that you can use. One is you can say, well, let's compare to what we already know and use reference sequences or reference library. Or you can take approaches where you say, well, I don't want to take any information from elsewhere. I just want to look at this, what we might call de novo, with a clear mind and a clear sheet of paper in front of me. When we're looking at reference-based phylogenetic profiling, there are a variety of different programs. You're going to be looking at Kraken. There are others called Metaflen2, Motus. The NCBI uses a program called STAT. And basically these will give you a tally of what organisms are present in what percentages in terms of relative abundance. You'll be able to visualize that in terms of pie charts like this one here. It's also worth stressing when we're talking about reference-based approaches that we can also use mapping, and you've done mapping before. The problem here is that if you want to map against everything that's known, that's in, say, the RefSeq database, that's going to take an awful lot of time and resources. But there are situations where you know what you're looking for. So if we're looking at the human gut and we want to see whether there's human DNA present and we want to screen that out, we can map against the human reference genome. Similarly, if you're looking at a chicken gut and you want to screen out the host, or you might want to screen out what's in the diet, like wheat, from your metagenomic analyses downstream. Reference-free phylogenomics relies on a process called binning. So we do the sequencing as usual, we do an assembly more or less as usual, like you do with a whole genome sequence, but what we then do is take those contigs and we try and bin those contigs into genomes based on various characteristics which we'll say more about later. And then later on, you can do mapping of those bins against the whole assembly or against references and so forth to do additional analyses. So in effect, what we're doing is we're taking all these little labels here, and we're trying to say here this is something I stole from Donovan Parks. You know, you've got the wombat genome and the koala genome, and you want to look at what organisms are present in one rather than the other, and what the relative abundances are, and you want to be able to see which metagenomes are more closely related to which other metagenomes. Now, one key concept that's used, particularly by Kraken, that we're going to talk about later, is the idea of k-mers. So what these are subsequences of the sequences in your sequencing library that have a length k that are contained within those biological sequences. So k just stands for a digit, a number. So we could have a 16-mer if we took length 16. We could have a 32-mer if we took length 32, and so forth. And these are fundamental units in genomic analysis for pattern recognition, sequencing and mapping. So if we take an example here, we've got that DNA sequence. As we run through it and chop it into 3-mers, we'd say, right, we've got ATC there. We move one along, and then we've got TCG, that's another k-mer, a 3-mer, and then we've got CGA and so forth as we move along. And in this way, we're defining all the possible 3-mers within that sequence. And typically one would count them and see how many examples of each of those 3-mers will be there. And that gives you a signature of the composition of that particular sequence. Generally, when you're doing these analysis, you set your k-mers at the same specific length of k. So you don't mix in 3-mers with 23-mers. You tend to do an analysis with one particular kind of k-mer. As we just seen, they overlap with each other. So the adjacent k-mers will share k-1 bases with the starting k-mer. And we can count each unique k-mer within a sequence, and that tells us about sequence composition and complexity. So if there are very few k-mers in the sequence, but it's a very long sequence, it shows it's a very low complexity sequence. Whereas if there are many, many k-mers, then this suggests that it's much a richer, more complex sequence. Now, you're going to be looking at a program called Kraken, in particular Kraken 2, the latest version. And this is a system designed to assign taxonomic labels to short DNA sequences, typically contigs, but can be used on reads, using k-mers. And it uses exact k-mer matches to known genomic sequences in a reference database. So, for example, if we know a particular k-mer occurs only in E. coli, then we see a match to that and we say, oh, we found a match to E. coli. And this system is very good because it's speedy and it's accurate, fairly accurate at least, in its taxonomic classification. And it's a heuristic approach. It's giving you a quick and dirty answer, not giving you the perfect answer, but it works quickly in real time, in a time that's suitable for most analyses. How it works is that Kraken builds an index of k-mers found in a reference set of DNA from known organisms. So you could take all bacterial genomes that you know, particularly people use the RefSeq database, which is a kind of curated set of bacterial genomes from NCBI, and you build a library, a Kraken library of k-mers from that. Then there's a classification algorithm, so when it analyzes a sequence, Kraken searches for each k-mer within its database, and then classifies by and assigns sequences based on the highest number of k-mer matches to a particular organism in the database. And so it's working its way through, and I think I've got some more graphical slides here. So you've got RefSeq genomes, for example, in a library that was used in this particular case, you've got 2,000 RefSeq genomes. Obviously, there are many, many more genomes now, and the problem with these analyses is they're only as good as the reference database used. So if you use a database made only of bacterial sequences, you're never going to detect the human genome, you're never going to detect fungal genomes. But if you try and use the largest possible, most complex reference data set, you'll make a very large library and your analyses will take longer. And I'm not going to go into the details of exactly how it works, but it's basically looking for the lowest common ancestor that shares that particular KEMA, to which that KEMA can be assigned. So a KEMA that might occur in Escherichia, in all Escherichia, um, um, it would be one example there. It wouldn't be discriminatory between Escherichia coli and another kind of Escherichia because it's found in all Escherichia. But there may be some that are found only in E. coli. And you can basically set the level of discrimination in terms of the hierarchy of taxonomy using this kind of approach. And again, this is another graphical picture just showing you how you have all these KEMAs and you map them to this lowest common ancestor, and you work out a classification tree and path, and KEMA will give you, the Kraken will give you the results based on these analyses of KEMAs. So just again to recap, we take metagenomes, we do shotgun sequencing, we get reads, we assemble them into contigs. We can bin them, but what we can also do first is look at the KEMA profiles, and that can be used sometimes in binning as well. But what we're going to particularly look at now is the broader picture of what you can do with all this information once you get there. So this is from a paper by Konstantinidis and his colleagues. And you start off with your metagenome, you assemble, you get contigs, you do binning. One of the key points that you have to then do, and we'll be doing that later in the week, is you check for completeness and contamination. So what you're trying to do is say all those contigs that have been coloured red because they share, say, a particular compositional signature belong in a bin together, they define a genome. But what you want to do is you want to say, well, does that represent the whole genome and nothing but the genome of the red organism? And what we look there for things like single copy marker genes that are conserved in all bacteria. Are they all present, our whole set? If they're all present, it means we've got a very complete genome. If they're only partially present, we've got an incomplete genome. Have we got multiple copies of some of them, which means we've got sequences coming in from another genome? Contamination. Then there's all these other things that you can do downstream of it, why it's useful to do this kind of stuff, phylogeny, genealogy, taxonomy, proposing new taxa, and doing functional analysis as well, all useful. So metagenomic binning is a process where we group together reads or contigs, usually contigs actually, from a metagenomic data set that belong to the same organism or closely related group of organisms. And there is some ambiguity here as to what we mean when we're binning. Ideally, we are actually trying to bin reads that belong in the same replicon, or even in the same genome. Obviously, plasmids and chromosomes, hard to bin those together, but ideally we'd like that. But often there isn't a single definitive genome sequence in that particular population for a particular organism. There may be slight variations. There may be more than one strain, and you end up binning reads from the same species, but from different strains together. So there is some ambiguity in how this proceeds and what our goal is. And what we want to do is reconstruct genomes from a complex mixture of DNA fragments. And obviously, but we want to get the equivalent of the genome sequence we would get from a cultured isolate, but without doing the culturing. And we don't want to do the culturing either because it's going to be impossible because this thing won't grow in culture, very difficult because it only grows very reluctantly, or sometimes this is the quickest way. If you've got 100 organisms in there, to tediously go through and pick colonies, even if you could pick colonies for 100 different organisms, it would take a huge amount of time. Whereas with metagenomics and binning, you can get out lots of information very quickly. And this allows us to study microbial communities and their functions in natural environments, including samples from humans and other animals, clinical samples. Now, how do we do it then? So there are various things that we can look at to say, oh, those belong together, those contigs, or those don't. So one thing is GC content. Typically, the genome has a kind of average GC content with a bit of variation around it. Things that share that GC content probably belong together. Codon usage is another parameter so some organisms will use particular codons rather than others. And oligonucleotide frequencies, which is effectively another name for k-mers, can also be used. So these are ways of grouping sequences by their composition. Another thing you can do, though, is if you've got multiple samples, you can do differential coverage binning, where you look at the abundance of genomes across multiple samples to segregate them into bins. So if you know that E. coli is present at 20x coverage in sample 1 and 200x coverage in sample 2, you can look at all those reads which, when mapped, or contigs which, when mapped against the whole assembly, actually give similar kinds of coverage. And so that's another way. And the other way is that you can use homology-based binning, although this is tends to be restricted only to these conserved single copy genes. So we use those for QC. You could also use homology-based approaches to refine your bins, but as a primary approach, it's going to be very labour-intensive. You're going to be using a program called Anvio, and this is an open source community-driven analysis and visualization platform for metagenomic and metatranscriptomic data. And what this allows you to do is to refine your metagenome binning approaches, where you can integrate multiple data types, and you can basically iteratively interact with the data to tidy it up and make it look cleaner. So it brings a human element in rather than you just accept what a pipeline gives you. And these interactive visualization tools also allow you to understand the communities better and their makeup. So it's got this multi-layered binning, so you can look at what happens if you predominantly bin by GC content or codon preference, what happens if you bin by coverage. You can explore it in real time, and there's plenty of community support for it as well. So that's just a very quick overview of some of the techniques you'll be using later in the week. You'll get more details from Andrea as he introduces each of the programs as we start using them. But that's just to provide the conceptual backdrop. In my own case, I've done lots of interesting work over recent years, looking at metagenomes in ticks with collaborators in Palestine, looking at what lives inside the chicken gut with collaborators at the University of Surrey, looking at ICU patients with collaborators in Birmingham. And I've even looked at what lives inside 200-year-old mummies and reconstructed TB genomes from 200 years ago using metagenomics. All of these approaches, in outline, in essence, very similar. What we're going to focus on this week is just one case study that provides a data set for you to look at. It's a clinically relevant data set. I think it's well anchored, so you'll find it rewarding to look at this data. Why do we care about patients on the intensive care unit? Well, these are the sickest patients in the hospital. They've got a wide range of patient needs. They're usually on ventilators, usually require intravenous parenteral nutrition, high use of antibiotics. They're very prone to infection. And what we know is that the gut microbiota in these patients acts as a reservoir of pathogens and antimicrobial resistance. And that we before we started the study, there was clear evidence that low microbial diversity was linked to adverse clinical outcomes. So basically, the gut acts as a reservoir where pathogens can hunker down and live. If there's antimicrobial resistance in those pathogens and you're giving antibiotics, those pathogens will bloom within that environment and then spill out from that compartment into other parts of the body, into the bloodstream, into the lungs and so forth. And so what we wanted to do was to just get some baseline data. What happens to the gut microbiota in these ICU patients? How does it respond to antibiotics? Do the antibiotics select for pathogens? And basically, what can we understand? What can we learn using shotgun metagenomics in a kind of brute force approach, just to take fecal samples from these patients and sequence them? And hovering in the background, is there a case for fecal microbiota transplants in intensive care unit patients? Could we restore a healthy microbiome to these patients and prevent them from getting sick and restore help them on their pathway back to health. So these are the individuals that helped us. Beryl Oppenheim, microbiologist, Tony Whitehouse, the ICU physician at the University of Birmingham, along with Willem van Schaik, who's a microbial ecologist there, myself and my postdoc Anu Ravi. And what we did is we took fecal samples from 30 ICU patients, we shotgun sequenced them, we did taxonomic reclassification with antibiotic resistance, and in some cases we achieved strain level resolution. So we wanted to say, oh, they've got this particular organism growing in their gut. What strain is that? And here is the clinical metadata on the patients that we looked at. You can see there's quite a variety of different backgrounds there. Some of these are patients who will have been into the intensive care unit after being fairly well fairly recently. So things like subarachnoid hemorrhage. In other cases, though, they will have had a long case of disease before they came in, and we'd expect them to have had a lot of antibiotics already. What we found, though, we were surprised at quite how extensive this was. 2/3 of the patients experienced low gut microbial diversity, and 1/3 of them what we called ultra low diversity, where their potential pathogens were blooming up to 10%, 20%, 30% more. And in 75% of the patients, we saw episodes where the gut was dominated by pathogenic strains. We did actually type the strains in some of these, and we showed that there were certain strains of Enterococcus faecium that were being transmitted within the ICU environment from patient to patient. They weren't necessarily causing disease at the time that they were being transmitted, but they were blooming in the guts and being transmitted from one patient to another. And this shows you the sorts of pictures we see over time. So typically a patient, if they've got a bloom of a pathogen, they will sustain that over several days. Sometimes it will get relatively better. Other times it will actually get worse over time. And as you can see in the bottom there with Enterococcus faecium, it comes and goes sometimes in a rather unpredictable way. In some cases, we saw multiple potential pathogens blooming in the environment. And you can see there above the antibiotics that we use. So most of these patients received multiple broad spectrum antibiotics. What we found among those antibiotics was one in particular, meropenem, had a significant effect on gut diversity. We did discover that many so-called beneficial commensals were lost during their time in ICU. We did sometimes see commensals bloom as well, and one of them in particular, Methanobrevibacteria smithii, which is an archaean, it's not a bacterium, was seen to bloom in many of our patients. Whether that is anything to do with the biology of what's going on, or whether it's just the last man standing after so many antibiotics, because archaea aren't sensitive to antibiotics, is unclear. But we did see severe loss of diversity and AMR pathogen domination in many of these patients. And there is a question of whether we should be doing a trial of fecal microbiota transplant in ICU patients. Similar trials are going on in very select subgroups, like patients having stem cell transplants in America. So what we found in this study was that shotgun metagenomics provided detailed information on microbiota trajectories and pathogens in ICU patients. We were pleased to find that we could actually interpret and make sense of the data. We saw clonal blooms of multi-drug resistant pathogens, and we could reconstruct genomes and provide insights into pathogen biology and epidemiology. And as I say, this is laying the foundation for further studies in the future, perhaps intervention studies. So that's just a brief overview of where we're at this week and where we're going to go. You're going to be looking at some of the samples from that study and seeing if you can find out what's in them, and then we can discuss what it might mean and what uncertainties there might be about the conclusions that you might draw. Okay,